Thank you so much. So first of all, it's a great pleasure to have you here. And I just wanted to tell you a bit more about the Allen Institute. I'm sure you know already quite a bit about it, but I wanted to tell you what makes it unique and why this is so important for us that you're here. So the Allen Institute started about 22 years ago now, and it was founded by Paul Allen and Jody Allen, his sister. Paul passed away a few years ago, and Jody is still the chair of our board. And our mission is to understand what makes us alive and how understanding biology can actually improve health and improve humanity. But we go about it a little bit in a different way. So as per the beginning, as per Paul Allen's mandate, we go after big quests, big questions in life sciences that couldn't be tackled by a particular laboratory or division. And so, for example, what are all the parts of the brain? It turns out there's 5,300 cell types in the mammalian brain, which is a lot considering the entire body has about 6,800. So the majority of diversity of cell types is in the brain. So to do something like this, let's map the brain, requires many people and a long-standing effort. And so we go at it with a different perspective. Teams of 80 to 100 people, multidisciplinary teams working together collaboratively, publishing together, and importantly, we share everything that we do openly. So all the data, but also the protocols, the metadata, and more recently, even the analysis tools. So someone could go into all this data, actually do analysis and publish a paper without the need to have or own a fancy microscope. And this is actually happening. There are more publications published by folks outside of the Allen Institute with our own data than inside. So for example, two publications that came out a few years ago have resulted in something like 130 now. So these releases of the data sets really have an exponential effect, an exponential impact in a field, and that's what we want to have. So, and these, you would say, well, many other scientists use this data to produce papers. But if we go younger and younger, where can we have impact? And where does, for example, removing access to fancy equipment as a barrier can actually lead, for example, undergraduates to make discoveries and to learn and explore real biological data. And so this is what we do. And at the moment, each of these big questions is either an institute, but we call it a moonshot. So it's about 10 years, sometimes 15, of concerted efforts trying to solve one of these problems. And I'll give you a little bit of a highlight of what we have here at the moment. So we have in brain the Allen Institute for Brain Science, which has a current moonshot, which is to map, let's say, the hardware of the brain. I mean, to put it simplistically, all the cell types and all the connections. And that's a lot. I told you just 5,300 cell types, but for example, one neuron can connect to 200,000 other neurons sometimes. So each of us has as many synapses or points of contact in our brains as stars in the Milky Way. Can you believe it? We carry around our own universe. To map that, helps us, for example, ask which are the cells more vulnerable in Alzheimer's disease or in other disorders or in depression. And then in the Allen Institute for Neural Dynamics, we look at how these cells behave, how all the neurons behave in the context of a simple decision. For example, should I eat or not eat, which is a decision I have to make every day. In the Allen Institute for Cell Sciences, we're trying to understand how cells go from being isolated and growing in 2 dimensions to actually forming an organ in 3 dimensions. And we're studying the simplest, a lumen-like imagine that would look like the intestine. How does that happen? You know, at one point, they start differentiating and shaping and forming a tissue. In the Allen Institute for Immunology, we're characterizing all the blood cell types of immune cells in teenagers, children actually, teenagers, young adults, and senior adults in healthy situations, but then also in cancer, in infection, a lot in autoimmune disease. And this is the largest data set ever published of immune cells that we just released this year. And finally, in collaboration with the Chan Zuckerberg Initiative and the University of Washington, we launched recently the Seattle Hub for Synthetic Biology, and that's a place where we actually engineer cells to record what happens in their environment in the genome. So these cells sense what's happening, and they have a recorder in their DNA that actually inserts the history. So at the end, if you sequence that cell, you know what happened to it. And we also fund projects outside of here through the Paul G. Allen's Frontiers Group at different universities around the world. So to give you a snapshot of the 20 years here, what you can see is, for example, the launching of the first version of the Allen Institute for Brain Science, and then the Mouse Brain Atlas, the Human Brain Atlas, and the Connectivity Atlas are released. This is what we mean by you launch a moonshot and then impact. Allen Institute for Cell Science, a few years later, the Allen Cell Explorer is released. These are all places and free tools where you can explore. The Seattle Alzheimer's Disease BrainSat Atlas just released. Allen Institute for Immunology, and then the Human Immune System Explorer, where all these data are. And we just launched this very comprehensive Allen Brain Cell Atlas, which has all these cell types I just told you about, but also the Alzheimer's disease cell types and what goes away first. So our goal is that people actually take these tools and not only use it for education, but even for discovery and anywhere. And we hope it also has an impact on people, on the next generation of STEM, but also on everyone. At the time where trust in science is at a low, being transparent, showing data with reproducibility certificates, having everything out in the open, how much it cost, what's the actual data, you can verify it. Don't trust me, trust the data. The data are there, are independently verified, go in, explore it. For everyone, we think these will have to happen more and more throughout, and we want to give the example. Now, this doesn't happen alone. We collaborate with many other, universities, for-profit, not-for-profit, foundations, and these are just the main ones where you could see some collaborations. But if I show you, this is more like it, in fact, and more and more every time. And maybe your institution or some of you will be up there soon. Sometimes one of these publications of an atlas has 30 institutions working together. This is also an example of collaboration and how teams can go further. To set up these collaborations, of course, it's a lot of work. And so at the beginning, things may go slower than we want them to go. However, we go further, and that is very, very important. Anyway, so this was what I wanted to tell you. Thank you so much for being here, and I think we have a little bit of time for questions if folks have questions, but if not, we can move along.
So everyone who's in the room knows I'm a microbiologist, so I have to ask the question, when is the Allen Institute for Microbiology going to open?
So we recently launched a call with NSF about what would be the impact of climate change in microorganisms, in fact. And we think and we hope it still will go through because it's a partnership where we put some funds. It's still probably not mature enough for us to start an institute, but we want to start mapping that world because, as you know, I mean, the majority of living things in the world, are micro, and they will be the most affected immediately by changes in temperature, changes in humidity, et cetera, outside and inside our bodies, right? So we have a lot of hopes in that adventure.
I also want to add as a follow-up questions. So how do you decide what will be the next institute to establish that you start from neural and then to immunology and synthetic biology? And first question is what would be the next step, and if and then how do you make the decision to go into a specific field? Thank you.
That's a great question. So typically it goes through a process where we involve the community. And, we have things like salons with many scientists and even larger charrettes and conferences where we listen to the community and what are the potential big questions out there. Now, we can't do them all. And so now, how do you choose, right? You would say, well, it's the one that would have the most impact. That makes sense from our judgment. But there are several aspects, and we actually internally have a process that I've been working with for some years, which we call the WHOW process: why, what, and how. And the why is this an important question? Is it now? I mean, are we ready to tackle it? And the last one that's important, is it us? Meaning, are we especially positioned to tackle these? Some things are great questions, and it's now, but we couldn't add much, right? We're not the experts. I mean, we could recruit experts. Some other things are right down our lane. So, for example, one of the next ones that we want to launch next year is a human brain accelerator that would characterize a lot of the normal human brain, but also the vulnerability in the major diseases. And human brain diseases now affect 3.4 billion of us, which is, a number that is hard to comprehend, even when you compare it to cardiovascular cancer. And so that's something that is needed now and where we are uniquely positioned to tackle.
You mentioned analysis pipelines and tools that you're making available. Can you speak a little more about that? Like what kind of tools do you have in the open? Like what can they accomplish?
Yes. So we'll start. There are tools to explore our data, and those could be of different types. Tools to explore images in the visualization. So a lot of things to analyze, segment images, classify things. And then tools more on the bioinformatics sides that would, for example, with sequences and single cell sequences would allow you to analyze which types of cells are where. In addition, we're now starting to experiment with things, for example, chatbots, where you could ask with natural language a question about, so you don't need to be, imagine, a bioinformatician. You could just type the question and get an answer. In addition, we have tools where now imagine you have your own data and you just want to analyze your own data using the same tools and map it to these atlases. So we have tools now, both in the imaging and the sequences, that are embedded. And our goal is these tools, a lot of the data we move to the cloud so people can access, and we want to put these analysis tools there. Some are already there, but more and more in the cloud, so that it's not like, oh, you have to download the data to your computer and then download some tool. You go there and you actually click and run the analysis. One of the experiments we're doing is in a publication from Cell Science this year, you actually can click in the figure of the publication and you go onto the data set and you have the tools to reproduce the figure or actually make a different figure if you want.
So this has been some really impressive work that is clearly putting technology forward. I'm kind of fascinated by the history of science, and there are examples of kind of great groups, kind of a social dynamic that is an agent of moving things forward. And I'm wondering if in the organization of your lab groups, if there are any models that you use to sort of organize the esprit de corps or environment of inquiry.
Yes, so it's an interesting thing. So I've been fascinated with the idea of creative institutions in the history of science. One thing is individuals, and we always pay attention to the individuals, but many individuals where those succeed also failed, and we don't tell the story of that. Many companies, when Google started, had ping pong tables and swings and didn't make it. So that's not the thing that makes you be the company. But we don't tell that story, right? We always tell the story from the narrative. And then we had ping pong tables and swings and creativity blossomed. So there are some interesting models, right? So, of course, the LMB last century, when there was the DNA, RNA in Cambridge is a great model, and the Sanger Institute, where, for example, Sanger himself was given 7 to 10 years to come up with methods to sequence proteins. And when they were reviewed, the colleagues hid Sanger because he had nothing at the end of 7 years, and they would actually have kicked him out. So they protected him, and then, at the end of 10 years, 2 major discoveries, 2 Nobel Prizes, not bad. Bell Labs is another place we look at. So places where there are creative, interdisciplinary teams of people that do these. And we look at the large projects in physics a lot. So, the CERN, of course, in particle physics, and the astronomy projects, because those are projects where to have access to the Andes telescopes or to the particle accelerator, you can't have access alone. And the community has to decide where are we going to point the telescope, right? I mean, you can't decide, I want it there. So there has to be a consensus, and then the data acquired is open, and many people use it. So there's a model that is more advanced a little bit. In life sciences, we now see more and more places using this team and open science approach for large moonshots. And so we hope that this will be the start of a new history in life sciences. And we continue to experiment internally on the ways to do science. And it's not easy, but I don't know if we have had a model for how to best harness these. I mean, it is relatively recent, just a few centuries that we have institutions dedicated to these. I would say even maybe 1 century, because before it was more the individuals, not so much the institutions.
Well, that is a fantastic question to
Thank
wrap
you.
up on. Thank you.
Thank you all. Have fun.